In 1928, a Peruvian archaeologist named Julio C. Tello was excavating burial sites on a remote peninsula on the southern coast of Peru. He had been working in the region for years, mapping the remnants of a pre-Incan civilization that history had largely forgotten. What he found in the desert sands of Paracas that season was unlike anything he or anyone else had encountered before. Hundreds of elongated skulls, not slightly elongated, dramatically, structurally, almost impossibly elongated, some of them nearly 50% larger in cranial volume than a normal human skull, wrapped in extraordinarily fine textiles, buried in a fetal position in underground chamber tombs cut deep into the desert floor. Tello understood immediately that these were not the product of post-mortem deformation. The bone structure itself had changed. The skull plates had fused differently. This was not a body modified after death. These were people who had grown this way, and the question of how and why they had grown this way would haunt archaeology for nearly a century before the DNA finally offered an answer that no one had predicted. The Paracas culture flourished on the southern coast of Peru between approximately 800 BCE and 100 CE. They are recognized today as one of the most sophisticated pre-Columbian civilizations in the Americas, primarily for their extraordinary textile work. Paracas textiles, many of them recovered from the same burial sites as the elongated skulls, are considered among the finest ever produced anywhere in the ancient world. The weaving techniques they used, the density of thread per inch, the complexity of the color patterns, the precision of the embroidery exceed what most modern handlooms can replicate. They also practiced something that was widespread but rarely taken to the extremes found at Paracas: intentional cranial deformation. Across many ancient cultures, from the Maya to various African peoples to certain European Bronze Age groups, the practice of binding an infant's head between boards or with tight wrappings was used to reshape the skull as it grew. The skull of a newborn is malleable. Under consistent pressure over the first years of life, it will grow in the direction that pressure allows. The result is a cranium that is elongated, flattened, or otherwise shaped according to the cultural ideal being expressed. At Paracas, this practice was taken further than almost anywhere else on Earth. The skulls are remarkable not just for their shape, but for their scale. Standard cranial deformation lengthens the skull, but does not increase its volume. The Paracas skulls are larger in total volume than any artificially deformed skull has a mechanical right to be. This is what sent the first wave of serious researchers down a path of increasingly extraordinary speculation. If the volume itself was anomalous, some argued, then perhaps what we were looking at was not deformation at all. Perhaps these people were simply born different. The theories that followed ranged from serious to spectacular. Genetic anomaly, an isolated inbreeding population with unusual hereditary traits, hybrid ancestry from a lost civilization, and then, inevitably, the theory that the internet would eventually make impossible to ignore: extraterrestrial origin, alien ancestry, the skulls as evidence of contact between ancient humans and beings from elsewhere. The alien hypothesis was never scientific, but it was not entirely without a hook, because the volume anomaly is real. The question is whether it is as anomalous as it appears, and whether there is an explanation that does not require rewriting the laws of biology. Before the DNA, the debate was largely confined to the morphological evidence, and the morphological evidence was genuinely puzzling. The Paracas skulls were studied by physical anthropologists who confirmed the unusual volume. They were examined by neurosurgeons who noted that the parietal plates, the bones on the sides of the skull, appeared in some specimens to show a different structural pattern than is standard in human anatomy, including what some described as a single parietal plate, rather than the two separate plates that all modern humans possess. This finding, if confirmed, would be significant. It would mean that whatever caused the unusual morphology was not purely the result of external pressure. It would point toward a genuine anatomical difference in the underlying bone structure. And an anatomical difference of that nature could only come from one place: the genome. The question of whether to pursue genetic analysis of the Paracas remains had been complicated for years by the same issues that complicated ancient DNA research everywhere in the Americas. Questions of indigenous consent, questions of national jurisdiction, questions of who has the authority to authorize the study of human remains from a culture that left no living community that clearly identifies as its direct descendant. The Paracas culture predates the Inca by centuries. Its genetic continuity into the modern population of coastal Peru, while likely in some diffuse sense, was not traceable through any specific living group. In 2018, a team working under the direction of researcher Brian Furster, in collaboration with a laboratory in the United States, extracted mitochondrial DNA from 5 Paracas skull samples and submitted them for analysis. The methodology was later scrutinized and debated in academic circles. The sample sizes were small. The chain of custody for the samples was questioned. The results were published not in a peer-reviewed journal, but in a series of public announcements that generated enormous popular attention and almost immediate academic skepticism. But the results themselves were striking enough that they could not simply be dismissed. The mitochondrial DNA analysis of the 5 Paracas samples returned haplogroup assignments that were unexpected. Most of them fell within haplogroups associated with Native American populations of South America, which was consistent with the archaeological context. But one sample returned a haplogroup designation that had no precedent in any Native American population. It matched most closely with haplogroups found in populations from the Middle East, the Caucasus, and parts of Eastern Europe. A single outlier result from a contested methodology is not a discovery. It is a question, and the scientific establishment treated it accordingly, with caution, with calls for replication under more rigorous conditions, and with the reasonable observation that ancient DNA contamination from researchers and handlers remains one of the most persistent and difficult problems in the field. But the question, once asked, did not go away, because the Middle Eastern and Caucasian haplogroup connection was not the first time that anomalous genetic signals had appeared in South American ancient DNA. Researchers working on entirely different questions, with entirely different populations, had been encountering something similar for years. The genomic data that changed the conversation came not from the Paracas skulls specifically, but from a broader ancient DNA study of South American populations, published in the journal Cell in 2021. The study, led by researchers from Harvard and several South American institutions, analyzed ancient genomes from across the continent, covering thousands of years of pre-Columbian history. Its primary goal was to reconstruct the population history of South America before European contact. What the study found, embedded in the genomes of certain ancient coastal and Amazonian populations, was a signal that the research team designated as Population Y ancestry, a genetic component that did not derive from the primary Beringian founding population of the Americas, a component that matched most closely with ancient populations from South and Southeast Asia, from the Andaman Islands, from lineages ancestrally related to the first inhabitants of Australia and New Guinea. This signal had appeared before in smaller studies. The 2021 Cell paper confirmed it at scale, with rigorous methodology and a large enough reference panel to rule out most alternative explanations. The conclusion was careful but clear. Some ancient South American populations carried ancestry from a source population that had not crossed into the Americas via the standard Beringian route. How that ancestry arrived was not specified. The data established the fact of the connection. It did not establish the mechanism. Here is where the Paracas skulls re-enter the story. Because the coastal Peruvian populations that showed the highest population Y signal in ancient DNA studies were geographically and temporally adjacent to the Paracas culture, not identical to them, not the same people, but close enough in space and time that the overlap could not be treated as coincidental without examination. If the Paracas people, or their ancestors, were part of a coastal population that had absorbed population Y ancestry at some point before or during the formation of the Paracas culture, then the anomalous haplogroup, result from the 2018 skull samples, was not necessarily the product of contamination or methodological error. It might have been a real signal from a real genome, a genome that carried ancestry from a source population that had arrived in South America by a route that no textbook had predicted. 3 interpretations have been seriously proposed, and none of them is fully satisfying. The first is the simplest. The anomalous results from the 2018 analysis were the product of contamination or methodological error. The Paracas skulls, like all ancient remains, are vulnerable to contamination by the DNA of researchers, handlers, and environmental sources. Without peer-reviewed replication under controlled conditions, the results cannot be taken as confirmed. This remains the position of most mainstream physical anthropologists. The second interpretation accepts the population Y signal as real and proposes that it represents very ancient ancestry carried by the founding population itself. Under this model, the people who first settled coastal South America carried population Y ancestry from a Southeast Asian-related source, acquired during the complex migrations that brought humans from Africa to Asia, to the Pacific Rim, and eventually into the Americas. The Paracas people, as a coastal population, may have preserved this ancestry at higher frequencies than inland groups, producing the apparent anomaly in the DNA record. The third interpretation is the most controversial and the least supported by current evidence, but the most interesting to sit with. It proposes that the population Y signal represents a contact event, that at some point before or during the Paracas fluorescence, maritime peoples from Southeast Asia or related Pacific populations made landfall on the South American coast, that they were absorbed into existing coastal communities, and that the extraordinarily fine textile traditions of the Paracas, which have no clear local antecedent in the archaeological record of coastal Peru, may represent cultural transmission from that contact as much as independent invention. This hypothesis cannot currently be proven, but the genomic data has made it impossible to dismiss as pure speculation in the way it once could be. What the Paracas skulls ultimately represent, stripped of the sensationalism that has followed them since their discovery, is a set of genuinely unresolved questions about human prehistory in South America. The elongated morphology, real and unusual as it is, most likely reflects an extreme but not biologically unprecedented form of intentional cranial modification, practiced by a culture that took a widespread ancient custom further than almost anyone else. The volume anomaly, carefully measured and genuinely surprising, may reflect a combination of the modification technique, post-mortem changes, and the particular genetic background of a population that was itself unusual in its ancestry. The DNA, incomplete as the current evidence is, suggests that the Paracas people were not visitors from another world. They were something more interesting and more human than that. They were the descendants of a convergence, of migrations and contacts and absorptions that played out over thousands of years along the Pacific coast of a continent that we are only beginning to understand in its full complexity. There is still no peer-reviewed ancient DNA study specifically targeting the Paracas skulls under rigorous modern methodology. That study has not been done. The political and logistical barriers remain significant. When it is done, the results will either confirm the anomaly, explain it away, or, most likely, complicate the question further in ways that no current model fully anticipates. That is how science works. Not with final answers, but with better questions. And the Paracas skulls, staring out from museum cases across Peru with their impossible elongated forms, are one of the most compelling generators of better questions that the ancient world has yet produced. They were never what the sensationalists claimed. They are something far stranger and more meaningful, evidence of a human past on the South American coast that is older, more connected, and more surprising than the history books have yet found a way to fully tell.